Then we come to the bioinformatics, and there I want to give you, first of all, an overview, so a path, what is commonly done for single-cell RNA-seq data, and quite often for other single-cell types. But if you do something else, then you might do additional steps or different steps. From the sequencer, you get raw data, a BCL file. At least for Illumina, it's definitely the case. These are your base calls. Quite often you don't have to deal with that. At least it never fell into my hands. I always got already the fast queues from the from other groups of the facility. However, if you have, for example, 10x Genomics, and then you can use CellRanger. They have a module called make fast queue, I think, or faster. And this module then creates from the base call files, so your raw data, your fast queues. I don't know for SmartSeq how you would do it now, but for 10x you can use CellRanger. From there, if you have the fast queues, and I now assume that everybody knows what that is. So it's basically your read file. Then you go and create your count metrics. I will explain what that means in a minute. And from this count metrics, you do your quality control. And in between count metrics and quality control, you do your ambient RNA filtering. At least this is how I know it now from CellRanger, that it uses the raw data and the raw data count metrics. And then you filter out your ambient RNAs and go into your quality control. After that, you do an another filtering where you remove emptyplets, doublets, and other things. And then you do then a data correction. Then your data integration and clustering. So each individual cell now, because we have high dimensional data, we need to bring each individual cell into a dimension so that we can later on use our data to then do the clustering to find distinct clusters, and then do our cell annotation. So this is also a common step. Doesn't have to be, but it is a common step to do, that you do then a cell annotation. So the clusters you identify, you then label them as specific cell types, for example, and then you do further third level data analysis. And another step is donor demultiplexing. So what I named now the donor barcode, multiplex barcode. So this now depends on your experimental design. So if you have this in your reads, then you also do an additional step where you do this donor demultiplexing. And I explain to that a bit more now in a few seconds. Now to the count metrics. So I will explain here CellRanger. This is one very known tool to use for 10x genomics, and all other tools, for example, like Salmon, they follow a similar structure. The first thing what they often do is read trimming, so they remove some primers, for example, and then you do your genome and transcriptome alignments. So your reads are aligned to your genome and transcriptome. With CellRanger, you use RNA star. Then you do a mapping quality adjustment, meaning you remove reads with a low base call quality. Then what CellRanger also does is a 10x barcode correction. So CellRanger also looks immediately for correct barcodes in your data. So it has kind of a whitelist of known 10x barcodes, and it looks, if it can find it in your read library, there is slight marginal, it allows a slight marginal error. So it allows for, I think, one or a Hamming distance of 1, meaning that there can be one base calling error to this barcode. And the fifth thing is then the UMI, well, deduplication and therefore the merging of the UMIs into one entity, and then the calling of the reads for your cell barcode. So basically the read pile ups, and at the end you get your count metrics. And how does this now look like? You will get a file which is called H5. This is a hierarchical structure format, and a specific format because single cell data can be quite big. And in order to handle this, this H5 format was introduced. And if you load this now in I'm quite often use Python, but you can also do it with R. Then you get an object called an undata, and this undata represents your count metrics, which you can see here in the presentation. And this count metrics, so you have integers, has now rows and columns, obviously. So it's 2D, and the rows, basically, if you ever deal with this undata, you then do operations, and quite often you do undata.obs, which stands for observations. These observations stands for your rows, and these observations are your cells. And then you have .var, which are your variables, and these variables can now be, for example, your genes. Yeah, your count metrics is basically the read count for your particular cell for this particular gene. And additionally, you have some unstructured or unstructured annotations in your undata, and these can be cell labels, QC, or sample IDs in there, which you put in there during your quality controls, and are useful for later on for your analysis or clustering. Yeah. Now let's come to quality control. I talked about the confounders, and one thing you quite often investigate for your individual cells is first the read count depth and the read count for your genes, which you see here in panel A and panel B. And I talked about that empty-plets, you have technically no cells or cells which are broken down, and therefore you have RNAs which then still sequenced but leads to low read counts for your genes. And these you can see here, for example, in this panel B, where you have a big peak around 1 to 2,000. These are your good cells, so to say. And then you have another smaller peak, which you can see, which is below 1,000. So I don't know, around 200, 300, or 100, 200. And these are basically your could be your empty-plets. And on the opposite side, your doublets can therefore be peaks which are on the higher spectrum. So where you see genes which way higher read counts. And the same goes also for generally for your read or count depth. You might also see these peaks, which you can see here, corresponding to your empty-plets and doublets. And for that, you can apply then, and I reference here this particular paper, and you might already, so if you already read about single-cell sequencing, then you came probably already across this paper of Fabian Theis current best practice. And they formulate that you can introduce a threshold to then filter out these empty and droplets. I have to point out here that over time, new tools came into existence which do a more sophisticated way in order to filter for that. So this whole setting up a threshold you can do. However, I have to. It's important to know that you then investigate not only the read count depths for the individual cells and the read counts for the genes, but also other things. For example, the mitochondrial counts, in order to find a good threshold so that you don't lose too much cells. So thresholds is always a bit dangerous between filtering out which you don't want, but filtering out not too much, obviously. And therefore, it's good to start with that and then try to use tools which does a more sophisticated method. Another point I want to make here is in panel C, you see the barcode ranks. And here you already see what I meant with in the literature, they always name now cells as barcodes. So these cells are here ranked based on their count depth. So the cell with the highest count is here on the left and with the lowest count here on the right. And if you see ambient RNA, then what you often observe is a little additional plateau here. And this additional plateau you can use then to filter out this ambient RNA. There exist now also tools like CellRanger, which make predictions about this background and then filters out these empty / ambient RNA droplets. Another thing to investigate is, I talk now more or less about quality control on the cell level. There's also quality control on the experiment level. If you apply CellRanger, then you get a report which looks like this. This might now, it looks a bit different if you do multi-omics, so there's, or single-cell ATAC-seq. So this is example for single-cell RNA-seq, and you can see then, for example, the estimated number of cells, which should correspond, obviously, to what you would expect from your laboratory work. So if this is very, very, very low, then something might have gone wrong with your experiment. Also, the number or the percentage of reads with valid barcodes and valid UMIs should be, well, basically high enough so that you have enough for later on for your data analysis. Also, what you can see are Q30 bases. So the bases with a quality Q score of 30 should also be in the Theis paper. They talk about, no, I have to remember, I think between 60 and 70%, it's okay. Above is obviously better. Below might be a problem, and you should investigate if this experiment is then worth the data analysis. And then additionally, you see also your mapping statistics, giving you an indication again about how well your experiment performed. So for example, reads mapped to intergenic regions, the percentage there gives you an idea if maybe you have some contaminations in your data, and then you can decide if you want to use the experiment or not. And additionally to these experimental controls, you might have other experimental controls based on the single cell type. Here you can see a plot which you quite often investigate for ATAC-seq or ATAC-seq. I always call it ATAC-seq. And you investigate there the insert size to see this particular pattern. I don't want to go now into too much detail because now you have to know what ATAC-seq is and how you perform it. What I just want to stress out is that additionally you would quite often then also perform quality controls that are specific for your single cell type, for example, this insertion size plot, and then you would decide if this experiment failed or not. So with that, I think I already now explained a bit more that after the QC, you do a filtering based on tools or thresholds for emptyplets, doublets, multiplets, ambient RNA, or experimental, or your experiment in general. And then we have the demultiplexing, donor demultiplexing. So short side or digression here. Why do I want to speak about this? Because I told you that we have a barcode for that. And technically it should be easy, right? So you have the barcode, you map the barcode to your patient, and then you know from which RNA your patient came from your RNA maps to your patient or individuum. However, there are also other methods to do the demultiplexing, and one thing which you nowadays sometimes see more often is genotype demultiplexing. So what do you do in there? So you don't have a barcode at all. So you have a pool of cells which come from a pool of individuals. And from your cohort, so your patient cohort, for example, you have obviously common variants, but also you can investigate which polymorphisms you have. So other variants you have for your individual patient, and based on this, you can estimate and generate your genotype for your patient. And with this genotype information, you can then look for your individual cells to which genotype this cell corresponds to. And therefore, you calculate a probability that then you can later use to assign the cell based on your variant information to a particular donor. So this is another possibility to do this demultiplexing. What I quite often ask, maybe somebody already tries to question then, is why should I do this? And so far, what I found in the literature is that it is more cost efficient and less, and also time efficient. This is at least what I found in the literature as an advantage. And this is technically a big thing because single cell can be quite expensive, as you can imagine, because you specifically if you want to investigate different individuals, because you have to generate a lot of, or you have to get a lot of cells which you want to sequence, and then for each individual patient. So this can be quite costly, and alone this introducing of this barcode can also complicate this read library creation, and therefore removing this barcode and doing this genotype demultiplexing might reduce costs and time. Then we come after the filtering to normalization. Also a common task in bioinformatics, which you quite often know, also from bulk, that obviously each individual cell have different total counts, and you therefore try to find size factors, which you then use for normalization in order to make the cells more comparable. In the past, you could use a lot of bulk techniques there, but nowadays there are also single-cell versions of normalizations, so which you have to use because you deal with quite sparse data, and therefore you have to account for this, and thus you have to rely on normalization methods which are optimized for single-cell data. There are also other ways to normalize, for example, with a spike in. One way to do it is you can introduce a reference cell in order to normalize your cohort. So you have you introduce a particular cell with a particular an A composition to your cell mixture, which you then sequence and use as a reference in order to normalize and in order to reduce the technical variance. Or you can use reference RNA, which introduce to every cell, and therefore this reference RNA, this count of this reference RNA, use them to normalize the cells. Another correction, which I already talked about, is the batch correction. And here on the left, you see what might happen if you have batch effects in your data. You see here there are some cells which more or less cluster together, which are introduced by technical variants. And these batch effects can be, so you can either already consider them by designing your experiment, so this balanced design, and then you can filter or correct this, which you here see on the right side, and therefore your samples should mix better generally. Then there is another step, and here I point out, be careful to use it. It's quite often wise to first do your data processing without it and your data analysis, and then try it out and see if this improves your later results. And this is something which is called imputation and smoothing. I mentioned that you deal with sparse data. You can imagine that one of the downsides of single cell is that you do not get much material from an individual cell. And the result is that you A, do a lot of amplification, which leads to duplicates, and B is that a lot of gene counts are small or nearly or completely zero. And this is the result, and you don't really know. So this is the result either of true expression, because the gene was not, or the gene was not active or repressed or whatever in your cell, or of technical variance, because it was for this particular cell not really captured. And one way to improve this is a smoothing or imputation, where you try to investigate how are the gene counts across all of my cells in order to infer the structure of the gene expression to fill up these zeros in my other cells. This can be done based on your current data, or you can actually also use reference data, so from a different cell atlas, to fill up these zeros, and therefore increase your yeah, to fill up your data. And I think that's the last one, I think, for data correction is cell cycle removal. And here again, caution to use it. Depends heavily on your biological question. Your cells obviously over time can be in different stages. And if you don't investigate, particularly now, the time course of sort of how your cells develop, and you, let's say, mostly interested about the cell types, then it can happen that your one particular cell type might actually split into 2 or more distinct clusters because your cells are in different cell stages. So you might know that you have transcriptional bursts based on these about the time course of your cell. And this you might want to take into consideration, and there are methods to remove this cell cycle bias. Again, be cautious to use it. If you're actually interested about the cell cycle, obviously you don't do it. And now we come to so almost to the last step where you go more into investigation. So here we are now into clustering. So basically data integration and clustering. I mentioned that your metrics and each individual cell is high dimensional. Also, we deal now with sparse data, so there's a lot of information which we don't really need. And what you try now to do is you want to learn, and this is often called latent variables from your data. So, and this goes into dimensional reduction. So that you can make these cells more comparable to each other. Else, there's something called the curse of dimensionality. Your cells would be hard to compare. And this you do in the data integration. So what you do is there the dimensional reduction, and then based on the reduced data metrics, dimensional reduced data metrics, you do your clustering. And based on these clusters, you then do your cell annotation, which you either already have based on a reference. So maybe there exists a paper which already studied this. And there's a reference data set which you can use to infer these cell types. Or you know that there you investigate specific cell types, and then you quite often have marker genes in order to infer these cell types. And yeah, one point I wanted to make, see this comes to my mind now, is there is a big difference between data visualization and data analysis and integration. So basically data integration and all the third level data analysis. What I mean is that for data visualization, and these are all the beautiful pictures you quite often see, you quite often do a different dimensional reduction and sometimes also clustering than what you would do for the data analysis. This has something to do with finding your latent variables and to basically define the distinct clusters. Now, the last part, and now I go into what can you do further. Obviously, there's now a wide spectrum what you can do, and I will cover here now 4 additional things you could investigate. First thing is compositional analysis. This means you investigate how are now your distinct cell clusters, so your cell composition in these distinct clusters differ between conditions, again, healthy, non-healthy, in order to understand the mechanistics behind a disease, for example. Second thing is trajectory analysis. There you investigate how the cells develop, or in which direction your cells develop. And this is an example where you would, for example, not do cell cycle removal, because you're interested in the time course of your cell and how your cell develops. And in trajectory analysis, you get then a prediction. This particular cell, not particular, doesn't have to be this particular cell type, but this particular cell in this particular stage goes now, might go into a steady state, for example, and is then stuck in there. Which you sometimes see for cancer, and then your cell will proliferate and so on. So you try to find a target, or and there you might have a target in order to stop the cell in going into the steady state. And this is what you can investigate with trajectory analysis. Third thing is the differential expression analysis. This is well known, also bulk sequencing, where you investigate different genes and see how is their expression profile pattern changing now between cell types, but also between patient or non-patient. So here it's also very important to note, of course, you can here also use methods which were already designed for bulk sequencing, but quite often nowadays there exist tools which are optimized for single cell data. And then the last one is gene set analysis, where you investigate, if you have now a set of differential expressed genes, investigate how do they link, which are they responsible for cell proliferation and so on. If you want to read further, here are again 4 papers I thought are very informative for these kind of things, so you can read up again. And with that, I would go into the last part. So I make this a bit more brief now. So this goes now into the direction what happens in GHGA, what products can you create, and there it's important to know that obviously you want to find a good standard now for the data analysis. And in GHGA, we try to make not only data fair, so maybe you heard about the fair data principles, so findable, accessible, interoperable, reusable, but also the data analysis should be fair, so the tools. And if you are new to bioinformatics, or even you are a bioinformatician, so you know the pain maybe already that we created a lot of monsters in the past, where we have very bad documentation, they cannot be run. If I run it, then the results I get are not reproducible. So in GHGA, we really try to make make the data we get fair, but also the data analysis. So meaning that we want to find a good standard that can be used in 10, 20 years. And we are working currently on a pipeline in Nextflow. So Nextflow is a workflow language. And there is the NF core. So this is basically the community behind Nextflow and the source base. And there is this workflow is currently for the basic data processing. So it will create your count metrics. It allows for SmartSeq2, 10x Chromium, and DropSeq. There are 4 different tools. One of them is CellRanger. And just briefly, so why Nextflow? So why should I use a workflow language? If you're not familiar with Nextflow, maybe you heard about SnakeMake. If not, well, CW is another prominent one. One of them is there's a good documentation for your workflows. So they're quite often well described, what they will do, what you get out of it, what you need as an input. There's continuous integration. So if you make changes to the workflow, so you implement new modules, then it's immediately tested if the workflow is still running. So also making sure that if there are new releases, that they are stable. There's also package software, goes again into software development. So if you heard Docker and Singularity, then you might know this is very useful for reproducibility and reusability. This helps with shipping your software from one computer/environment to another and make it runnable, and runnable in a way that you can reproduce your data, your results. And that's what package software stands for. Yeah, so portable, reproducible, and Nextflow is also able to make the workflows cloud ready. And the future direction of the single-cell workflow is that we want to integrate also single-cell ANAseq and multi-omic. We are currently working on it. If you're also happy to contribute, so feel free to contact me if you want to work together with me on this pipeline. However, there is, you saw after the count matrix, there comes also more stuff. And this is covered by another pipeline, a bioinformatics pipeline called scFlow, where you have quality control, dimensional reduction, clustering, and so on. We still want to test this and further develop this pipeline. But obviously, there are also other pipelines out there. And now the last part I want to talk about is a bit how can we make sure, and this goes now into the question, what open challenges do we still have in single cell? How do we make sure that the tools like a pipeline or a general tool for a specific task, which currently is the best, stays also the best? Or we decide later on to move to a different tool. And there exists now, which I really find useful, I want to stress it out here, community and the web page called the Open Problems in Single Cell Analysis, which try to create benchmarks or benchmark sets in order to rank and in order to optimize individual tasks for single cell data analysis. And I will skip now these things because of time. But check this out if you want to contribute to if you want to create tools and contribute to the single cell community. And the last slide I want to just mention. So here's a big list of literature, which I already had in the presentation, but there are also more literature in here for guidelines. Meaning it's a paper where you have a list of tools or descriptions how to do your single cell data analysis. I want to also point out one GitHub repository from Sean Davis, which is very useful, the awesome single cell. So it's a GitHub repo where you find another big list of tools for individual tasks, some papers. So check this out if you want to read more and explore more. Here's a list of tools and the two pipelines which we use in GHGA. And here are papers for individual benchmarks, which you might want to check out. So here you have some collection for reading up after my webinar. And with that, I thank you very much for your attention. Also, of course, to the Nextflow community, GHGA, and the PIs, which are basically also involved in the single cell community. Yeah, thank you very much.